So we are doing things a little out of order, and we are talking about concept 1 from our heredity unit, and we'll get into that in a little bit. But I like to talk about DNA structure and its function and replication during the cell cycle because DNA is replicated during the cell cycle. Also, it makes life a lot less confusing if you're not talking about DNA and RNA at the same time because they are so similar. Even though they have a lot of similarities, they're quite different. Hopefully you're looking at this picture on the page and some of this is review. You're like, oh yeah, I remember that term, or I know that shape, or I know those dots mean hydrogen bonds. And so let's delve a little deeper into this. Okay, you know that DNA is a nucleic acid. The Na reminds you, nucleic acid. And a nucleic acid is just a type of macromolecule, and it is made up of nucleotides. Those are the monomers. And the macromolecule of nucleic acids, their job is to contain genetic information. And then that genetic information is used to make proteins, and proteins make up every part of you: skin, hair, muscles, a lot of parts of your cells, that sort of thing. The other thing that DNA does is it controls the cell cycle, and it will signal it's time to make new cells. There are two types of nucleic acids that we've talked about: deoxyribonucleic acid and ribonucleic acid. You need to write that down. You need to know that DNA stands for deoxyribonucleic acid. How did it get its name? Because the sugar in it is named deoxyribose, so it's named after the sugar. At the bottom of this page, it says, where do we find DNA? You know this, and it says it in your sidebar. It's always found in the nucleus. Now, from our reading yesterday, we did know that it's also found in a little bit in mitochondria and chloroplasts. So I like this picture. I think it's actually on your next page, but it's just kind of like DNA is found in the nucleus of the cell, and then it is in chromosomes. The chromosomes are in have little segments. The segments are genes, and the genes are simply just nucleotides of DNA. Just a segment of those nucleotides all put together, maybe 50, maybe 500. Okay, you really need to know the different parts of the nucleotide. So it always has sugar, phosphate, and a nitrogen base. Those three things every time. The sugar, if it's RNA, is ribose. If it's DNA, it's deoxyribose. And then you have these little, and they're usually represented by circles. It's phosphate. It's basically phosphorus and 4 oxygen. We call it phosphate. The sugar and the phosphorus make up the backbone. And then there are 5 nitrogen bases: A, T, G, C, and then U. We find T, thymine, only in DNA, and U, uracil, only in RNA. They're very, very similar. So what's the structure of DNA? You've been hearing this for a while now. It's a double helix, or sometimes we call it a twisted ladder. Now, if you can picture a ladder in your mind, it has sides and it has rungs. The sugar phosphate make up the backbone, or the sides, and the nitrogen bases, when they pair together, they make up the rungs. And they always pair in the same pattern, A to T and C to G. So we call these the complementary base pairs. And they are fit together perfectly like puzzle pieces. So look in the sidebar, you see a picture of RNA because it's only half of a molecule, so it's single-stranded. You've got this sugar phosphate backbone, and my question to you is, how many nucleotides are in this picture? And the correct answer is 4. So you have a phosphate, a sugar, and a T, that's one nucleotide. A phosphate, a sugar, and a G, that's another one. A phosphate, a sugar, and an A, that's another one. A phosphate, and a sugar, and a C, that's the 4th one. There's also one phosphate that's just kind of hanging out there that doesn't have a nucleotide to be a part of right now. So if you look at the picture right below it, you actually see a double helix. And so they have these little dotted lines there. And those are hydrogen bonds, and they hold both sides of the ladder together, and they're relatively weak. That's a good thing. That allows us to easily break them down and separate them so that we can replicate them. And we use each side as a template. More on that in just a little bit. But my question to you now is, in this picture of DNA that we have, how many nucleotides are there? And the answer to that are 8. There are 8 different nucleotides. So the base pairing rules: adenine A always binds with thymine T, and cytosine C always binds with guanine G. Remember in the Amoeba Sisters video, we had a little saying to help us remember: apples in the tree, cars in the garage. I really like that. But I used to teach at Curious George's Hiking the Appalachian Trail. If that helps you too, go for it. So we want to do a little practice. So here is the strand of DNA. I don't have the whole nucleotides. I'm just got the letters represent the nitrogen bases. You'll see this a lot because the sugar and the phosphate stay the same all the time. So what I want you to do on your paper, pause this, is try to figure out the complementary bases. So I'm going to talk you through the first 3. So you have T. T is going to bond with A. So right below the T, I'm going to write an A. And then the next one is A. Right below A, I'd write a T, because A always bonds with T. And then G, what is it going to bond with? C. All right, you keep going, do the rest of it, and then check your work. You can pause it here. If that's what you came up with, great. If you didn't, look at it for a little bit and see if you can figure out why that is the way that it is. All right, so we talk about DNA, chromosomes, chromatin, and I want to take a little bit of time to make sure that we really understand it. So chromosome and DNA mean the same thing. Just a chromosome is a good bit of your DNA. Your DNA in humans is organized into 43 different chromosomes. And the chromosome is the DNA, and it's like coiled around itself, and then coiled around itself some more, and coiled around itself some more, and it finally gets into these pretty little X's that we call chromosomes. So I said it likes to coil around itself. It needs little things to help keep it straight. We call those things histones. They're just a special type of protein. Now, if you were to uncoil it and look at a segment, we call that segment a gene. More on that in 2 slides. But what I want you guys to do is to fill in the blanks. So we've kind of clarified what happens to the DNA during the cell cycle. So DNA during interphase, and when it's just resting doing its thing, it's coiled and it's in chromatin form, the form that looks like spaghetti. During the S phase, the chromatin is going to coil some more and form those pretty chromosomes. That makes it easy for them to pull apart during metaphase, anaphase. Then during telophase, they're going to relax back into that chromatin. We call that being uncondensed. So they're not going to be the pretty X's anymore. They're going to look like spaghetti. The reason that they do that is it actually allows them to be packed up smaller. It's like if you kind of smush everything into a drawer, sometimes you can fit more stuff in there rather than if you like were to fold it nicely. Although I do like my things folded nicely. Okay, so I told you we'd come back to genes. So a gene is a small section of DNA that codes for one particular protein. And that protein is going to make a trait. So for example, I have dry skin that cracks. So in my DNA, it tells my body to make the protein for my skin, and my skin happens to not make that protein very strong, so my skin will crack in dry weather. It's not very fun. I also have a section of DNA that tells my body when it's making the hair protein to make it curly. So it just depending on the organism, how many genes there are in there. The proteins are usually over hundreds of the nucleotide bases, which is crazy. Okay, so during the S phase, we replicate DNA. And it has to be replicated so that each new cell has the right DNA. Are you enamored by the little GIF there? I love it. So you need to make a copy so that everybody has a set of instructions. So both cells that we're making have a set of instructions. And it's going to happen during the S phase. And we call it DNA replication, and it's making an identical copy of itself. And it's just to make sure that everybody has an exact set of chromosomes. I know I've just said this twice. Okay, so the nucleotides must be copied. There's no place to write this down right now. And they copy in complementary ways. So they split the DNA, that double helix molecule, in half, and then they use the base pairing rules to match the new nucleotides. And it's really easy to split the DNA in half because the hydrogen bonds that hold the two rungs of the ladder are easily broken. And so then each strand serves as a template. It says, make this, make the opposite one. And I love this little diagram up here. We have the blue one, and then the blue splits into two, and the yellow is the new. So we call it semi-conservative replication because the blue is saved, so it's conserved, and the yellow is new, and half of it's old and half of it's new. So step one to do this, helicase unzips the DNA. Helicase ends in ACE, so that tells us it's an enzyme. So there's an enzyme that comes in and breaks down those weak hydrogen bonds. Then this big, huge enzyme named DNA polymerase finds the place where helicase has unzipped it, usually because there's something called DNA primase that comes in there. You don't need to know that. And it says like, it's like a air traffic controller. It says, come here. And the DNA polymerase is going to then be a builder, and it builds the new DNA molecule. And then finally, they kind of get recoiled with their new one, and that's done by an enzyme called lyase. When we're finished, it's semi-conservative because they have one old strand and one new strand. Sorry. You have half as old and half as new. Sorry about that. All right, after we're done with this, we're going to start working on our DNA models, which are on page 6 in your packet. And it tells us how to color them, and we're going to make one giant model for the class. It's awesome.